Hi, Dr. Wang here. Today I'm going to first do a short lecture on PCR, and then we are going to go over assignment PCR 1. So PCR is a molecular technique. It stands for polymerase chain reaction. It's actually very similar to replication, right? So in replication, which happens within the cell, we start with one copy of the genomic DNA, and then it's replicated that produce two copies. So PCR is very similar to that, but this can be carried out in a test tube. So I'm going to start with this double-stranded DNA template over here. I'm going to call this the DNA template. So in the middle of this DNA template is a region that we want to amplify in the test tube. So PCR has 3 steps. The first step is called denature. Denature is when we raise the temperature to close to like 95 degrees, so 95 to 92 degrees. And then what this does is that it's going to denature the double-stranded DNA. It's going to break open the double-stranded DNA so that now they look like this. In DNA replication, there is a similar step just like that, right? So please try to recall which enzyme is responsible to break the 2 strands of the DNA into single strand. What is that enzyme? And then that is the answer that you're going to use in question number 4. All right, so after that, we are going to lower the temperature. So we usually lower this to somewhere between 50 to perhaps 60 degrees. So this step is called annealing. During this step, what's going to happen is that a small DNA called the primer is going to now be able to bind to the template, right? So the bottom strand is running from 5 to 3 in this direction. So the primer, usually only 20 nucleotide long, is going to now form base pairing with the template's DNA and forming a structure like that. So similarly, annealing to the top strand, we should have a primer that goes this way. I'm now going to label the 5' and 3' direction of this primer. So the reason that the primer can now anneal or bind to the DNA template is because we lower the temperature. This allows the DNA primer to bind to the template. So also let's think about during replication, which enzyme is responsible for producing the primer, right? The name of that enzyme that produce primer, that again is the answer for question 4. So as we discussed during DNA replication, DNA polymerase cannot start polymerization from scratch. So it always requires a pre-existing polymer. In the actual replication process, that is an RNA primer produced by the cell. In the test tube, of course, we do not have that enzyme. So instead, we provide the primer. Right? This primer is actually can be synthesized by companies. We simply ask company to synthesize the primer that we need, and then we add that primer to the PCR mixture, so they can anneal to the template DNA. So this is step number 2, annealing. Right, so 1, 2. So the third step is called extension. So extension normally occur at temperature that's roughly 32 degrees. So here we use an enzyme that's called tag DNA polymerase. So this is an enzyme first discovered in microbes that lives in Yellowstone hot springs. So it's highly resistant to high temperature. Very few enzymes can perform at such high temperature, but tag DNA polymerase can. So the tag DNA polymerase is going to come in and add nucleotide to this 3' hydroxyl group, right? So this is what DNA polymerase does. It's going to add one nucleotide at a time to the 3' hydroxyl group. So it's going to do this to both here, and also it's going to extend the 3' end of this primer as well. So the polymerization reaction is going to run this way and then run this way all the way to here. All right, so now I'm going to basically draw out the structure, right? So this is the first cycle of the PCR. We are going to get a product that looks somewhat like this. So here, because this primer is at a relatively 5' position, so we often call this primer either the forward primer, right? So because it's going in the same direction as the top strand, we either call that the forward primer, or sometimes we call it the 5' primer. And then this primer over here, we call that the reverse primer because it's going in the reverse direction. And we sometimes also call that the 3' primer. So this is the first round, right? So 1, 2, 3, 3 steps, cycle 1. So because this is carried out in a test tube, we can actually control the reaction temperature. So we can actually come back and then go back to step 1. So what's going to happen here is now denature will occur again. So because after cycle 1, we have 2 double-stranded DNA. So in cycle 2, both 4 strands that we have here, all 4 strands can now serve as the template to start another round of DNA replication. So normally in a normal PCR, we usually repeat this 30 cycles. So we can imagine, right? So if we start with a single double-stranded DNA molecule, after 1 cycle, we are going to get 2. Right, so and then after another, we're going to get 4, and then it's going to double. So the amount of DNA molecule doubles after each round, right? So 1 cycle, we have 2 to the power of 1, 2 cycle, 2 to the power of 2, 2 to the power of 3. So after 30 cycles, we are going to get 2 to the power of 30. This is a very large number. So this is why it's called a chain reaction. Chain reaction implies that this is a reaction that's basically taking on an exponential growth. So as you can see here, we start with 1 molecule, we end up with 2 to the power of 30. This is a very large number. So this is a way of producing basically a region of the DNA, we can produce large quantities in a test tube in a very short amount of time. So PCR is used very frequently nowadays. For example, the COVID-19 test, part of it is a PCR test. It's going to amplify part of the viral genome so that we can detect that on the gel. So the product of this, so let me just draw that, right? So I'm going to say after 30 cycle, what we are going to get is something like this. So the forward primer, right? So, and then the newly synthesized DNA right here, and then the reverse primer right here, and then the newly synthesized DNA right here. So this is the double-stranded structure that's going to be produced at the end of the PCR cycle. So we can see, I'm going to highlight that on the original DNA. It's basically from the beginning of the forward primer all the way to the end of the reverse primer. So this is the region of interest, a region that can be amplified by PCR. So coming back to what we're going to do in the lab today is that we are going to basically amplify a region from the PMSH2 plasmid, because in the last lab, we already determined that sample B and sample C are either PMSH2 or PMSH2 prime. We do not know which one is which, right? So what we're going to do in this lab is to use a pair of primer. One goes this way, the other goes this way. All right, so similarly here, it's going to go here and it's going to go here. So what this is going to do is to amplify a region. I'm going to highlight this region, the beginning of the forward primer all the way to the end of the reverse primer. So this is the region that's going to be amplified by PCR. Similarly here, we're going to amplify a region that is right here. So this is a smaller fragment. So that's a lot easier to analyze later on. So the only difference here is that there is a mutation right here in PMSH2 prime, right? So here we have a mutation that can be amplified by this PCR reaction. And next week, we are going to do additional tests on those two amplified PCR fragment in order to distinguish PMSH2 from PMSH2 prime. All right, so now coming back to this assignment right here. So we went through the definition and the process of the PCR reaction, and you actually should have everything all the way to question 4. So the only tricky question is probably question 3. So for this, I would recommend that you actually watch this video that's embedded in here. All right, so I'm going to come back because question number 5, this is not a PCR question. This is a review question on the 3 repair mechanism and the DNA replication process. So for this, please pull out your CMAP. Please pull out your CMAP for DNA repair mechanism, the 3 mechanisms, and also pull out your CMAP for DNA replication. What are all the enzymes involved in DNA replication? And that will allow you to answer this question.